Your daily dose of wellness begins now.
From leprosy to cancer. Another hot take with Dr. Nathan Goodyear here at Prescribe. Now, I got to give credit to my colleague, Dr. Jason Williams at the Williams Cancer Institute, because he found this article. And this article really, I think, expands the awareness and the vision of the power of repurposed medications to be the bridge between, the kind of natural holistic approach that so many of us want and that patients desire, and the conventional power, but provide them in a safer manner. And it's really interesting because when you look at this concept of this repurposed medication, it's actually a repurposed medication for leprosy. So really, we're also bridging time. We're bridging Bible to modern times as it relates to the treatment of cancer with a repurposed medication. Now, of course, this repurposed medication wasn't available back in the biblical times, but leprosy obviously is talked in the Bible. And here, this repurposed medication approved for leprosy actually is showing its power in immunotherapy as a repurposed medication. Now, the title of this article is Clinical Drug Screening Reveals Clofazimine Potentiates the Efficacy while Reducing the Toxicity of Anti-PD-1 and CTLA-4 Immunotherapy. Now, so just a little bit of background. When we talk about conventional cancer care, we're talking about most therapies, chemotherapy, radiation, surgery, immunotherapy, targeted medications. It's a systemic to tumor approach. And of course, at the Williams Cancer Institute, we go to the tumor to activate the immune system. And the problem with going through the body to get to the tumor is all the pharmacokinetic limitations, liberation, absorption, distribution, metabolism, excretion that can limit the delivery, the saturation, and the intended effect of the drug in the tumor, but also in the tumors, because often we have multiple tumors involved. And when we talk about immunotherapy from a conventional standpoint, really we have to talk about front and center immune checkpoint inhibitors. These are really what brought to the public consciousness the awareness of immunotherapy. And immune checkpoints actually serve a purpose to protect the body. See, we need the immune system basically going through the body, identifying all enemies, foreign and domestic. We don't need it attacking self. And immune checkpoints allow the immune system to be regulated, to monitor, so that it doesn't attack self. And PD-1 and CTLA-4 are two such immune checkpoints. Now, PD-1 inhibitors are programmed cell death protein 1 inhibitors. Now, these are found on T cells. These are receptors. They bind to ligands that cancer cells will upregulate, PD-L1, PD-L2, and this then interaction between the receptor on the T cell, the PD-1, and the PD-L1 or 2, what that will do is that will then send an off signal to the T cells, thus deactivating them. And T cells are an immune activating cell that we need to fight to target cancer. In fact, much of what we do in the immunotherapy world is to activate T cells against cancer. And so by doing so, what you do is you prevent immune activation in targeting the tumor, both locally, but then also systemically. And when you look at immune checkpoint inhibitors, what they do is they actually block that interaction between the receptor and the ligand. And that then allows T cell activation, proliferation, and then immune activation against the tumor. Well-known PD-1 inhibitors would be nivolumab, pembrolizumab, and cemiplimab. Now, just a side point, whoever comes up with the names of these drugs, they should be tarred and feathered. It's ridiculous. It's a Scrabble, bonanza with these drugs. Then the CTLA-4 inhibitors, which is cytotoxic T lymphocyte antigen 4 inhibitors, they also are another immune checkpoint, and these are also on T cells. Now, they compete competitively with the stimulatory receptor, what's called CD28. And these are interacting then with the antigen presenting cells. These are immune cells that come along. They engulf the dead and dying pieces of the cancer cell. They then process it, basically then deliver that and present that with major histocompatibility complexes to the rest of the immune system to then activate throughout the rest of the body. And so here, CTLA-4 also again on T cells will dampen the immune system response. So by coming in and blocking it, what you're then allowing it to happen is CTLA-4 activation, proliferation, and anti-tumor activity. So this is the concept of immune checkpoints, which are good, but here cancer doesn't create anything anew. What it does is it hijacks these so that it can invade the immune system. Now, when you look at systemic immunotherapy, the response is modest at best, and it's really not very good. When you look across many meta-analysis on the topic, you're looking at about 15 to 20% any response rate. meaning there's any positive response. Now, when you combine the two together, the PD-1s, the PD-L1s, and the CTLA-4s, then that's where you can actually see, like in melanoma, an overall response rate of about 40 to 50%. So you can actually see these combinations together, the stacking of these conventional immunotherapies, and you can actually see this impact elevate significantly. So I mentioned the melanoma combination therapy. It's actually 50 to 60%, not 40 to 50%. Non-small cell lung cancer, it's 35 to 40% in combination. Renal cell carcinoma, 40 to 42%. So what you're seeing is there's a significant upregulation overall response in the combination compared to the low teens to low 20s when you just simply are using monotherapy. Now, the problem with this, and oh, by the way, on the CTLA-4, ipilimumab, which is a drug well known, it is a CTLA-4 inhibitor. The problem with the combination of these is the side effects, the adverse events, skyrocket. And so we have a grading system, what's called the common terminology criteria for adverse events to grade these adverse events. There's grade 1 through grade 5. Grade 1 would be mild, basically minimal or no symptoms. Grade 2 is moderate. These require minimal intervention, but they're not going to interfere with daily activities and definitely not going to require a hospital admission. Grade 3 is severe, medically significant, not life-threatening, maybe hospitalization. But then grade 4 is life-threatening. It's urgent, immediate risk to life. Of course, hospitalization required. And grade 5 would be the ultimate adverse event, which is death. Now, this study was looking at the ability to enhance the efficacy of anti-PD-1 and CTLA-4 combination therapy by also reducing the side effects. So a combination objective, improve efficacy and reduce side effects. Win-win for the patient. And they scoured about 3,000 FDA-approved drugs to try to identify a therapeutic. And here's where they came up with the Clofazimine. Again, traditionally used as leprosy. It's also been repurposed in tuberculosis. But here it was combined with the anti-PD-1 and the CTLA-4 inhibiting therapy. And what they found is that the addition of the clofazimine to this dual immune checkpoint inhibiting therapy significantly increased the anti-tumor immune response across various cancer models. They looked at colorectal cancer, they looked at melanoma, they looked at lymphoma and lung cancer. And in one mouse model, particularly looking at colorectal, they found that the combination, that is the two immune checkpoint inhibitor therapies plus the clofazimine, it actually led to an 80% tumor-free survival rate, which was better than the combination alone. So it really upregulated the efficacy tremendously above the two alone, which obviously we said that it stacks and it potentiates itself in that therapy. But here was the exciting thing. See, because when we talk about these interventional therapies, we always have to worry about the toxicities. The clofazimine actually reduced the severity of the adverse events reported here. Now, when we look at the combination of these therapies, you're going to see neurological toxicities. You're going encephalitis. You're going to see myocarditis. You're going to see colitis. GI issues are a major, major adverse event associated with systemic immunotherapy. In many cases, they lead to the reason why the therapies are themselves stopped. And then that brings me to the point of how does conventional medicine handle these adverse events associated with not the monotherapy, but the adverse events associated with the combination therapy to try to improve that efficacy, but you get these systemic side effects. Well, they do really kind of two things. Number one is they reduce the dose. And in a way, that's good, but you could potentially reduce the efficacy. Number two is they, through protocols, add in steroids to suppress the immune response. Now, when you think about it, it's like, okay, I can see that, but you're actually intentionally undermining the anti-tumor immune activity that you're actually inducing with the stacking of these two immune checkpoint inhibiting therapies. So it really is kind of a quandary when you go, what? So we reduce the dose, reduce efficacy because you can't tolerate the side effects, or you add in a steroid, which reduces the side effects, but it reduces the immune response, which is your whole objective of the entire therapeutic strategy. But what they found is that the addition of the clofazimine, it maintained a robust anti-tumor response, and it reduced the severity of the side effects. So just one repurposed medication, one which you've probably not heard of, It's not your run-of-the-mill, it's not your protocol, but here it's actually augmenting the immune system. Now, it's augmenting immunotherapy by augmenting the immune system. So here we look at the mechanisms of action. There's a couple. One is primarily looking at CD8 T cells. Now, CD8 T cells are a effector T cell that the immune system needs and we need in the anti-cancer strategy to target tumor cells. What's happening here with clofazimine, a little technical, it's actually promoting the activation of E2F1. And what's important about this is this plays a critical role in the elimination of self-reactive immune T cells. And it plays a regulatory step in the G1 to S phase transition. These are the growth phases here associated with these T cells. And in so doing, by activating these E2F1s and CD8 T cells, this led to increased transcription of what's called TCF7 and increased expression of TCF1. Now, all of that translates to this: increased T cell development, T cell differentiation, T cell function, T cell activity, anti-tumor activity, maintaining optimal immune function in an anti-tumor way, because there's a battle ongoing constantly in the immune system in the tumor microenvironment in patients with cancer. It's a battle between pro-tumor and anti-tumor. And here, the CD8 T cell activation, because of clofazimine, is actually tilting this immune system battle, one from pro-tumor to anti-tumor. And this is important because this also creates T cells that have memory. This is not just a treatment for the immune system that's abrupt and acute. We want a sustained time. We need treatment now, but we need immune activation later. And that's where T cell memory also brought on by a part of clofazimine addition to this combination therapy. Now, then we come into another mechanism of action is the modulation of Th17 cells. Now, Th17 cells are often upregulated in cancer because they create an immunosuppressive environment. What's interesting about Th17 is they produce interleukin-17, a cytokine that suppresses the immune system, creates an immunosuppressive environment. This allows cancer to evade the immune system, a significant rate-limiting step in how cancer spreads. But here, the interleukin-17 produced by Th17 promotes tumor growth through that immune suppression, invasion. It promotes angiogenesis, blood vessel supply growth. It inhibits apoptosis, which is programmed cell death. And what's also very striking about Th17 is it has plasticity. It means it can modulate to a T-regulating cell, which also further creates immunosuppression. And so we have the ability to counteract these Th17 cells, even T regulatory cells and interleukin-17, through the addition of clofazimine here, basically upregulating the anti-tumor environment and actually reducing the immunosuppressive environment. So basically creating an immune homeostasis, but one that is anti-tumor. And of course, if you want to go deeper dive on this, I encourage you to check out the show notes for this hot take, because the references will be there. Now, so here's the conclusion. To increase the efficacy of these immune checkpoint inhibitors in combination, to activate the immune system, you really have to play off the side effects. And many of those patients cannot tolerate. And this is where the fear associated with these medications come in, because you can get some significant immune responses, overall responses here. Now, we need better than what's provided in these studies, for sure. But if we can provide these therapeutics in a much safer strategy that improves quality of life, then that's what we need to be after. And here, repurposed medications stand up front and center. They raise their hand and say, count me in. And clofazimine, an antibiotic used for leprosy and tuberculosis here, stands up to actually improve the efficacy of the combination therapy, but actually reduce the toxicity. And so overall, it's going to allow the patient to receive the therapy, tolerate the therapy, and move forward. See, this is the power of an integrative strategy. We build bridges between natural, holistic, integrative, and conventional, because the goal here is optimal outcome and high quality of life. And at the Williams Cancer Institute, we seek simultaneous objective of elimination of the tumor through the activation of the immune system and to improve the quality of life. You don't need to try to get through therapy to survive and then live and then heal and then recover to then live. You can begin your living today. So thank you for joining me on this hot take. I'm Dr. Nathan Goodyear, and we will see you on the next hot take.
Thanks for joining us on this episode of the Prescribe Podcast. All references supporting this episode can be found on the prescribe.com website. That's pre-scribe.com. To get your next dose of medical innovation, make sure you subscribe to Prescribe. We'll see you for your next refill.